All right, welcome to your Phylum Porifera notes, or as they're also called, the sponges. So a quick overview of Porifera. So these are the most primitive of the multicellular animals, and this is also why we start with this phylum first. They also have no tissues or organs. They're made of a collection of cells, and there's actually some debate if they're not actually protists instead of animals. But for now, we still consider them classified as an animal. Sponges usually occur in shallow water habitats, and they can vary in size. So they can be up to 1 meter high, and their shapes also vary as well, which we'll talk about later. They're also sessile filter feeders, which means they don't move. And there's over 7,000 species. 40 of them are actually in our local waters off the coast of Georgia, and only 2% of all sponges are freshwater sponges. And none of them are terrestrial. So here's just a quick look at some of the diversity. So you see a rope sponge, a tube sponge, and a black ball sponge.
Good morning.
So in terms of anatomy, this is a look at the overall anatomy. So it's really important to realize that water comes into sponges from the side, and it comes out through the tube. So all of that incoming water that you see goes in through all those pores, and it all comes out this way. So up here is where it comes out. And just a look over here, if you look at the simple anatomy, so it shows the inside, that central cavity. It also shows the different cells, the pores, the collar cells, and we'll get into those in a little bit later. So the cell types. So pinacocytes are those thin, flat cells that are on the outer surface, and some of them can also be specialized into tube-like cells called porocytes. So porocytes specifically create the pores that connect the inside to the outside, and that's where that water goes through to do that water circulation for the sponges. Mesenchyme cells are also important because they can specialize to other functions. So they're almost kind of like a stem cell for a sponge. And things they can do include reproduction, they can secrete skeletal elements, they can transport and store food, and they also form those contractile rings around the openings in the sponge wall. Here's a look at the choanocytes. So these are collar cells is what they're known as, and they basically act as a pump. They bring water into the sponge, they circulate the water while it's in the sponge, and they help with the currents in general. So here's a look at mesophyll, which is a collagen, and it's found between the canals and chambers. It's basically all of that space that's in between those cells. And something else called amoeboid cells. These are located in the mesophyll as well, and these are very similar to a stem cell too. So you have the archaeocytes, which are amoeboid stem cells, and sclerocytes, which create spicules, which we'll talk about in a moment. So skeleton types. There's 3 major types. So sponges can be spongin, where they're soft and flexible, kind of like the bath sponges that you might be familiar with. There's spicules, which make them feel prickly, and then sponges can actually also have both. So they can be soft and flexible, but when you squeeze them, you feel the prickly part of the sponge. So here's a look at the support. So if you see, a lot of these cell types are labeled. So we have the archaeocytes here. The mesohyll is all of this space, all the dotted area, that's all the mesohyll. Then you have your porocyte, which creates something called the ostium, which is where that water flows in. And then you have the pinacocyte, which is on the outside, and then these random shaped structures are all spicules. And then let's not forget our important ones, the collar cells. So here's a look at spicules in general. So the collagen is stiffened when spicules are added, and they're usually a mineral or a protein, or they can be a combination of both. So the spongin itself will have that, but the spicules are going to be skeleton-like structures, and they're made of calcium carbonate or silicon dioxide. So they're going to be harder, and they're going to give that sponge more structure and support. So here's a look at water currents. So the currents that come into the sponge are bringing food and oxygen. The currents that are leaving the sponge are going out through that opening. Those are taking metabolic and digestive waste. So something important to note too, since sponges are filter feeders, their type of food filtration actually determines their body form. So there's three types of body forms or body plans. So there's the Sycon, Sycon, and Leucon, and those are going in order from left to right. And they also go in order from simple on this end to complex on this end, and you can easily see why that would be the case. So the first one is the Sycon body plan, and this is the simplest, like I just mentioned, but it's also the least common, and it's basically just a simple tube. So water enters the ostia and it leaves the osculum. So the ostia are all these little spots here, and the osculum is that big opening at the top. The Sycon body plan is more chambers, so it's not just that simple structure. And the choanocytes line the canals instead of just sponge cells. So if you remember, the choanocytes are your collar cells. So you have this structure here. If you see, it's very zigzag, and it's all lined with collar cells, and their little tails help the movement of the water as it's filtering through the sponge. But once again, water does come in through the ostia and it goes out through the osculum. So Leucon body plans, these are the most complex, and they're highly folded and twisted. If you see here, this inner space in the middle is very different looking from the previous two body plans. And the water is going to move from the canals and chambers, and there's actually multiple exit points. It doesn't just exit through the osculum. And it's important to note that there's no spongicell present, and the spongicell is that big area. So feeding of porifera. So porifera are actually pretty unique in their feeding. They can eat bacteria, algae, protists, suspended organic matter, but they're really important as well. They reduce the turbidity or the cloudiness of coastal waters, and in places that are having issues with coral reefs, they're actually seeing an increase in the turbidity. And a simple sponge, it's important to note, can filter 20 liters of water per day. Just one single simple sponge that's very tiny. So that's essential to the health of our coral reefs and our oceans. And how this happens is just that trapped food hits those collar cells, it moves to the bottom of the sponge, and the sponges have something called a food vacuole. So stimuli, they don't have nerve cells, they actually have nerve reactions, and these are from individual cells responding to a stimulus. And some of them can actually be restricted by light, because high light restricts that pore constriction, kind of like your eyes, if you think about how that functions. So there's also several reproduction types of sponges. So we have asexual, which is just budding. So they just branch off another part right off their body, and that's the new baby. Regeneration, if let's say a hurricane or something would go through a reef, those pieces can regenerate to form whole sponges. Survival pods are something unique to sponges. They're unspecialized cells, and those remain dormant until the conditions improve. So once again, if there was a natural disaster or something that happened, once the waters are safe for the regeneration or the regrowth of a sponge, those cells will regrow into a sponge. And then sexual reproduction. So sponges are hermaphroditic, also known as monoecious. So they have male and female cells throughout their bodies. But they can't self-fertilize. So the eggs and sperm are produced at separate times, intentionally, to make sure that they're not self-fertilizing, that there actually is that exchange of DNA between different sponges. And these are produced during meiosis, as you would expect. So the sperm cells are going to exit and the egg cells remain. And here's just a look at what that looks like. So this circle here is the important part where we start. It's zoomed in, if you see the little circle there. So it's showing the egg cell, and then it's showing the sperm coming out and going to that egg cell to fertilize. So then it will eventually form a larva, and that larva is going all the way over here. So it'll be actually able to move at that point. And then once it attaches to the surface, it becomes sessile as well. And that's where your new sponge develops. So after fertilization, like we said, the neighboring cells are fertilized. But the early development actually occurs in the mesohyl. So they're not going to just fertilize and then be released into the water. They are going to develop a little bit inside of the sponge where the egg was located. And what happens is you get these little larvae. They don't have mouths though. So they spend about 2 days swimming, and then they're going to settle where they grow into an adult sponge. So here's just a look at that life cycle. Once again, we have a male and a female where the sperm is going. And remember, every sponge can be male or female based on when the eggs are released and when the sperm are released. So then fertilization's here. You have that larva, and you're eventually going to go to that young sponge that plants itself somewhere to start growing. So here's just a look in detail of the different cells. If I were you, I'd pause this here. You may want to sketch what some of these cells look like. You will be responsible for knowing and recognizing the different types of sponge cells. The other thing we're going to talk about is shapes. So porifera have three common shapes. They can be vase-like, tree-like, or encrusting. And there's also three classes of sponges. So class Calcarea, Demospongiae, and Hexactinellida. So Sclerospongiae also used to be a type of sponge, but it's no longer considered a class. So Calcarea is the class that has those spicules. So these are going to be very hard sponges. They're not squishy like you would think about your bath sponge. And they're mostly small in size, typically less than 15 centimeters. And they form irregular masses. Like I said, they never contain spongin, so they're not going to be that squishy that you would think of, and they're restricted to shallow water and strictly marine. Class Demospongiae has those spicules that are made of silicon dioxide, spongin, or a combination of both. So these are the ones that when you look at them, you think they're squishy, and then you squish them and they have some sharp parts to them. Most sponges actually belong in this class, and nearly all of them are that leucon body type. And they're mostly found on the continental shelf, and this is just an example of the bath sponge. The last one is the Hexactinellida. So these are your glass sponges, and these are not very common. So they have spicules that are made of silica, and they're usually found in deep water on those soft substrates in the tropics. All right, sorry about the bell there. And spicules are six-pointed, and they have a lattice-like structure, as you can see down here. It's very evident in what these sponges look like. And they can be cup, vase, or urn shape. And that is all for your notes.